Hello everyone! Today we will be discussing cellular respiration, a series of metabolic processes by which cells convert biochemical energy into ATP. Cellular respiration begins with glycolysis for all cells. Here's a quick overview of the process. It is an anaerobic process, meaning that it does not require oxygen to occur. Glycolysis literally means the splitting of sugar, and that is exactly what it is. The breakdown of glucose into two pyruvate molecules. In other words, one 6-carbon molecule is divided into two 3-carbon molecules. This process occurs in the cytosol and is composed of 10 individual reactions. In the end, we gain 2 ATP, or energy, and 2 NADH. NADH is a coenzyme which is present in all living cells and acts as an electron carrier in cellular respiration. So let's take a closer look at it. So here we have one glucose molecule. 2 phosphates are stripped off of 2 ATP and they become 2 ADP. The phosphates attach themselves to the ends of the glucose molecule, creating a fructose molecule. Then, the fructose molecule is divided into 2 pieces. One by one, 2 NAD pluses come and take the hydrogens from the 2 pieces to produce 2 NADHs. Then, as 2 more phosphates attach to the ends, DHAP and G3P are created. Then, one by one, 4 ADP are converted to 4 ATP as they take the 4 phosphates from the DHAP and the G3P. And here we have 2 pyruvate molecules. But before we move on to the Krebs cycle, we must go through pyruvate oxidation. In this process, the pyruvates are converted to acetyl-CoA, which is essential for the Krebs cycle. This occurs in the mitochondrial matrix for eukaryotes and in the cytoplasm for prokaryotes. Carbon dioxide is released in the process. Also, the 2 NAD pluses are reduced to 2 NADHs. We have the pyruvate making its way over to the mitochondrial matrix. The NAD plus is reduced to NADH as the hydrogen is taken. Our carboxyl group is removed, while an acetyl group is transferred to coenzyme A. Meanwhile, carbon dioxide is also released, and we are left with acetyl-CoA. Finally, we have the Krebs cycle, named after its discoverer, Hans Krebs. It is also called the citric acid cycle because the first product of the reactions is citrate. Like pyruvate oxidation, the Krebs cycle occurs in the mitochondrial matrix in eukaryotes and in the cytoplasm in prokaryotes. The Krebs cycle has 8 major steps, as we will soon see. For each glucose, the Krebs cycle produces 3 NADH and 1 FADH2, both electron carriers that will later be used in the electron transport chain, as well as 1 ATP. During the process, carbon dioxide is released. So here's citrate, and here we have 1 NAD plus taking a hydrogen and becoming NADH. As this is done, 1 carbon is released as carbon dioxide. Now, we see this happen again. Next, with a phosphate, 1 ADP is transformed into ATP. One FAD becomes 1 FADH2, an electron carrier similar to NADH. Another NAD plus becomes NADH, and we are left with oxaloacetate, a 4-carbon acceptor. Next, we have the electron transport chain. This is a series of proteins, both transport and enzymes, that are built into the intermembrane space in the mitochondria, which is called the cristae. It is the last step of aerobic respiration. The process yields 36 ATP from 1 glucose molecules, as we will soon see. Here we see the electron transport chain. The electrons and protons of NADH split and the hydrogen is transported across the membrane. Meanwhile, the electron is transported to the other end of the chain. This happens twice. Then the same happens with FADH2. In the end, the hydrogens are transported to the other side of the membrane through ATP synthase. ADP is converted to ATP. Then water is formed. And we are done. I hope you guys enjoyed the video. Thank you for watching.